not only plasma B cells. Plasma B cells, they will produce antibodies which will go and bind to this. Then it is presented to the antigen presenting cells, macrophages ಅಂತ ನಾವು ಹೇಳ್ತೀವಿ ಇದಕ್ಕೆ ಆಂಗ್ಲ ಭಾಷೆಯಲ್ಲಿ. ಇಲ್ಲಿ ಮೆಮರಿ ಬಿ ಸೆಲ್ಸ್ ಕೆಲಸ ಏನಂದ್ರೆ ತದನಂತರ ಎಷ್ಟೋ ಒಂದು ವರ್ಷದ ನಂತರ ಮತ್ತೆ ಅದಕ್ಕೆ ಯಾವುದೋ ಒಂದು ನಿಜವಾಗ್ಲೂ ರೋಗವನ್ನ ಉಂಟು ಮಾಡುವಂತ ಈ ಒಂದು ಕ್ರಿಮಿಗಳು, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರ್ಬೋದು, ವೈರಸ್ ಇರ್ಬೋದು, ಇದು ದೇಹದ ಒಳಗಡೆ ಸೇರ್ಕೊಂಡ್ಬಿಟ್ರೆ ಆವಾಗ ಮೆಮರಿ ಸೆಲ್ಸ್ ದೇ ಗೆಟ್ ಟ್ರಿಗರ್ಡ್ ಇಮಿಡಿಯೇಟ್ಲಿ. They will start making several such antibodies, and then these antibodies will trigger the highly virulent infectious microbial population including bacteria or virus against which the vaccination is already carried out. ಇದು ಒಂದು ಸಮಾಚಾರ. ಇಲ್ಲಿ ಇದು ಆಂಟಿಬಾಡೀಸ್ ಇದ್ರ ಜೊತೆ ಮಾಡಿಕೊಂಡು ತದನಂತರ ಇದು ಮ್ಯಾಕ್ರೋಫೇಜಸ್ ತಗೊಂಡು ಮತ್ತೆ ಮ್ಯಾಕ್ರೋಫೇಜಸ್ ಅಲ್ಲಿ ಇದ್ರದ್ದೆಲ್ಲ ಪ್ರೆಸೆಂಟ್ ಕೆಲವು ಪ್ರೋಸೆಸಿಂಗ್ ಎಲ್ಲ ಆಗುತ್ತೆ. ಮತ್ತೆ ಮ್ಯಾಕ್ರೋಫೇಜಸ್ಸು ಅದು ನಿಮ್ಮ ಬೇರೆ ಬೇರೆ ನಿಮ್ಮ ಸೆಲ್ಸ್ಗೆಲ್ಲ ಪ್ರೆಸೆಂಟ್ ಮಾಡುತ್ತೆ. ಅದು ಒಂದು ವಿಚಾರ. ಇನ್ನೊಂದು ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಈಗ ನೋಡಿ, ಈ ವ್ಯಾಕ್ಸಿನ್ ಅಲ್ಲಿ ಇರುವಂತಹದ್ದು ಇದೆಲ್ಲ ಒಂದ್ಸರಿ ದೇಹದಲ್ಲಿರುವಂತಹ ಜೀವಕೋಶಗಳಲ್ಲಿ ಸೇರ್ಕೊಂಡ್ಬಿಡತ್ತೆ. ಒಂದ್ಸರಿ ದೇಹದ ಒಳಗಡೆ ಜೀವಕೋಶದಲ್ಲಿ ಸೇರಿದ ನಂತರ ಇಲ್ಲಿ ದೇ ಆರ್ ಆಕ್ಚುಲಿ ಗೋಯಿಂಗ್ ಟು ಬಿ ಕಿಲ್ಡ್. ಈ ಜೀವಕೋಶಗಳಲ್ಲಿ ಯಾವ್ದ್ರಲ್ಲಿ ಆಲ್ರೆಡಿ ವೈರಸ್ ಇದೆಲ್ಲ ಸೇರ್ಕೊಂಡ್ಬಿಟ್ಟಿದ್ದೇವೆ, ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಕಿಲ್ಡ್ ಬೈ ದೀಸ್ ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಈ ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್ ಎಲ್ಲಿಂದ ಬರುತ್ತೆ ಅಂದ್ರೆ ಇಲ್ಲಿ ಆಲ್ರೆಡಿ ಹೇಳಿದ್ನಲ್ಲ, ಬಿಳಿ ರಕ್ತ ಕಣಗಳಲ್ಲಿ ಇದು ಹಳದಿ ಬಣ್ಣದ್ದು ಟಿ ಸೆಲ್ಸ್ ಅಂತ, ಅದು ಒಂದು ಬಗೆಯ ಒಂದು ವೈಟ್ ಬ್ಲಡ್ ಸೆಲ್ಸ್. ಇದು ಏನ್ ಮಾಡುತ್ತೆ, ಫಸ್ಟ್ ಇದು ಹೆಲ್ಪರ್ CD4 ಟಿ ಸೆಲ್ಸ್ ಅಂತ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಈ ಹೆಲ್ಪರ್ CD4 ಸೆಲ್ಸ್ ಇದು ಸೈಟೋಕೈನ್ಸ್ ಅಂತ ಕೆಲವು ನಿಮ್ಮ ಕೆಮಿಕಲ್ಸ್ ಅನ್ನ ರಿಲೀಸ್ ಮಾಡುತ್ತೆ. ಈ ಕೆಮಿಕಲ್ಸ್ ದಿಸ್ ವಿಲ್ ಟ್ರಿಗರ್ ದ ಕಿಲ್ಲರ್ CD8 ಟಿ ಸೆಲ್ಸ್ ಟು kill these cells of the host system which are already infected by the virus. So that is the reason, because these white blood cells are going to kill these cells which are infected by the virus or the bacteria, hence the name killer T cells. ಇಲ್ಲಿ ಯಾವ ರೀತಿ ನಾನು ಜ್ಞಾಪಕ ಶಕ್ತಿ ಇರುವಂತಹ ಕೆಲವು ಬಿ ಸೆಲ್ಸ್ ಕೂಡ ವೃದ್ಧಿ ಆಗ್ತವೆ, ಅದೇ ಪ್ರಕಾರ ಟಿ ಸೆಲ್ಸ್ ಅಲ್ಲೂ ಕೂಡ ನಮಗೆ ಮೆಮೊರಿ ಟಿ ಸೆಲ್ಸ್ ಅಂತ ಕೂಡ ವೃದ್ಧಿ ಆಗುತ್ತೆ. ಯಾಕಂದ್ರೆ ಸಬ್ಸಿಕ್ವೆಂಟ್ಲಿ ಒಂದು 6 ತಿಂಗಳ ನಂತರ ಅಥವಾ 1 ವರ್ಷ ನಂತರ ಮತ್ತೆ ಈ ಲಸಿಕೆಯನ್ನ ಪಡೆದಿರುವಂತ ಒಂದು ಮನುಷ್ಯರಿಗೆ ಅಥವಾ ಪ್ರಾಣಿಗೆ ಅದು ನಿಜವಾಗ್ಲೂ ಹೈ ವಿರುಲೆಂಟ್ ಇರುವಂತಹ ಒಂದು ವೈರಸ್ ಅಥವಾ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇನ್ಫೆಕ್ಷನ್ ಮಾಡಿಬಿಟ್ರೆ ಆ ಸಂದರ್ಭದಲ್ಲಿ ಮೆಮರಿ ಟಿ ಸೆಲ್ ಎಲ್ಲ ಇದ್ದಿರೋದು ಇಮ್ಮಿಡಿಯೇಟ್ಲಿ CD8 ಟಿ ಸೆಲ್ಸ್ ಎಲ್ಲ ಆಗುತ್ತೆ. ಮತ್ತೆ ಇದು ಹೋಗ್ಬಿಟ್ಟು ಈ ಜೀವಕೋಶಗಳಲ್ಲಿ ಯಾವ್ದಾವ್ದಕ್ಕೆ ಆಲ್ರೆಡಿ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿ ಹೋಗ್ಬಿಟ್ಟಿದೆ ಅದಕ್ಕೆಲ್ಲ ಸಾಯಿಸ್ಬಿಡತ್ತೆ. ಇದು ಈ ಒಂದು ವ್ಯಾಕ್ಸಿನ್ ಯಾವ ರೀತಿ ಕೆಲಸ ಮಾಡತ್ತೆ ಅಂತ ಒಂದು ವಿಚಾರದ ಒಂದು ಚಿತ್ರಣ. ವಾಟ್ ಆರ್ ದ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ವ್ಯಾಕ್ಸಿನ್ಸ್? ಈಗ ಬೇರೆ ಬೇರೆ ಬಗೆಯ ಲಸಿಕೆಗಳೆಲ್ಲ ಯಾವ್ದ್ಯಾವ್ದು ಅಂತ. ಈಗ ಲೈವ್ ಅಟ್ಯೂನೇಟೆಡ್ ಅಂದ್ರೆ ಈಗ ವೈರಸ್ಗೆ ಅದಕ್ಕೆ ಜೀವ ಇರುತ್ತೆ, ಬಟ್ ಅದಕ್ಕೆ ರೋಗ ಮಾಡುವಂತ ಶಕ್ತಿ ಇರೋದಿಲ್ಲ. ಯಾಕಂದ್ರೆ ಅದಕ್ಕೆ ರೋಗವನ್ನ ಉಂಟು ಮಾಡುವಂತ ಶಕ್ತಿ ಅಂದ್ರೆ ವಿರುಲೆನ್ಸ್ ಅನ್ನ ನಾವು ಲ್ಯಾಬೊರೇಟರಿಯಲ್ಲಿ ಪ್ರಯೋಗಶಾಲೆಯಲ್ಲಿ ಅದನ್ನ ಸಾಯಿಸಿರ್ತೀವಿ. ರೋಗವನ್ನು ಉಂಟು ಮಾಡುವಂತ ಶಕ್ತಿಯನ್ನ ಮಾತ್ರ ನಾವು ಸಪ್ರೆಸ್ ಮಾಡಿರ್ತೀವಿ, ಸಾಯಿಸಿರ್ತೀವಿ. ಹಾಗಂತ ವೈರಸ್ ಅನ್ನ ಸಾಯಿಸಿರೋದಿಲ್ಲ. ವೈರಸ್ ದೇಹದ ಒಳಗಡೆ ಹೋದ ನಂತರ ಇನ್ನೂ ಅದಕ್ಕೆ ವೃದ್ಧಿ ಆಗುವಂತ ಒಂದು ಶಕ್ತಿ ಕೂಡ ಇದ್ದೇ ಇರುತ್ತೆ. ಅದಕ್ಕೆ ನಾವು ಅದಕ್ಕೆ ಲೈವ್ ಅಟೆನ್ಯೂಟೆಡ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಇನ್ನೊಂದು ಇನ್ಆಕ್ಟಿವೇಟೆಡ್ ವ್ಯಾಕ್ಸಿನ್. ಇದರಲ್ಲಿ ವೈರಸ್ ಅನ್ನ ಸಾಯಿಸೇ ಬಿಟ್ಟಿರ್ತಾರೆ. ದೇಹದ ಒಳಗಡೆ ಈ ವೈರಸ್ ಅನ್ನು ಹಾಕಿದ ನಂತರ ಅದಕ್ಕೆ ಮತ್ತೆ ವೃದ್ಧಿ ಆಗುವಂತ ಅದೊಂದು ಲಕ್ಷಣ ಕೂಡ ಇರೋದಿಲ್ಲ. ಈಗ ಕೋವ್ಯಾಕ್ಸಿನ್ ಅಂತ ನಾವು COVID-19ಗೆ ವ್ಯಾಕ್ಸಿನ್, ಅದು ದಟ್ ಈಸ್ ಆಕ್ಚುಲಿ ಇನ್ಆಕ್ಟಿವೇಟೆಡ್ ವ್ಯಾಕ್ಸಿನ್. ದೆನ್ ಟಾಕ್ಸೈಡ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಈಗ ಟಿಟಾನಸ್ ತಾವೆಲ್ಲರೂ ಕೂಡ ಕೇಳಿದಿರ. ಟಿಟಾನಸ್ ಅಂದ್ರೆ ಕ್ಲಾಸ್ಟ್ರಿಡಿಯಂ ಟಿಟಾನೆ ಅಂತ ಆ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾದಿಂದ ಉಂಟಾಗುವಂತ ಒಂದು ರೋಗ. ಕ್ಲಾಸ್ಟ್ರಿಡಿಯಂ ಟಿಟಾನಿಯಿಂದ ಟಾಕ್ಸಿನ್ ಉಂಟಾಗುತ್ತೆ. ಟಾಕ್ಸಿನ್ ಅಂದ್ರೆ ವಿಷ. ಆ ವಿಷವನ್ನು ಕೂಡ ನಿಷ್ಕ್ರಿಯ ಮಾಡಿಕೊಂಡು ಆ ವಿಷವನ್ನು ಕೂಡ ನಾವು ವ್ಯಾಕ್ಸಿನ್ ತರ ಬಳಸಬಹುದು. ಅದಕ್ಕೆ ನಿಷ್ಕ್ರಿಯ ಮಾಡಿರುವಂತಹ ಟಾಕ್ಸಿನ್ಗೆ ಟಾಕ್ಸೈಡ್ ಅಂತ ಹೇಳ್ತೇವೆ. ದೆನ್ ಸಬ್ ಯೂನಿಟ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂದ್ರೆ ಒಂದು ವೈರಸ್ಸಿನ ಒಂದು ಭಾಗನ ಮಾತ್ರ, ಇಡೀ ವೈರಸ್ ಅಲ್ಲ. ಅದ್ರಲ್ಲಿ ಸ್ಪೈಕ್ ಪ್ರೋಟೀನ್ ಇರುತ್ತೆ, ನ್ಯೂಕ್ಲಿಯೋಕ್ಯಾಪ್ಸಿಡ್ ಎಲ್ಲ ಇರುತ್ತೆ. ನಾವು ನಮ್ಮ ಈ ರೇಬಿಸ್ ಕಾರ್ಯಾಗಾರದಲ್ಲಿ ತಮ್ಗೆಲ್ಲರೂ ಕೂಡ ನಾವು ರೇಬಿಸ್ ಒಂದು ಚಿತ್ರಣ ಎಲ್ಲ ಕೂಡ ತೋರಿಸಿದೆ. ಇಲ್ಲೊಂದಿದೆ ನೋಡಿ, ಇದು ಒಂದು ಇನ್ಫ್ಲುಯೆನ್ಸಾ ವೈರಸ್ ತರ ಇದ್ರೆ ಇದು ಎಲ್ಲ ಸ್ಪೈಕ್ ಪ್ರೋಟೀನ್ಸ್ ಇದು. ಅಥವಾ ಇದು ಕೊರೋನಾ ವೈರಸ್ ಅಂದ್ರೆ ಸ್ಪೈಕ್ ಪ್ರೋಟೀನ್. ಕೇವಲ ನಾವು ಸ್ಪೈಕ್ ಪ್ರೋಟೀನ್ ಮಾತ್ರ ತಗೊಂಡು ಅದನ್ನ ನಾವು ಲಸಿಕೆ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಅದಕ್ಕೆ ನಾವು ಸಬ್ ಯೂನಿಟ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಹೇಳ್ತೇವೆ. ದೆನ್ ಕಾಂಜುಗೇಟ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಕಾಂಜುಗೇಟ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂದ್ರೆ ಕೆಲವು ಇದರಲ್ಲಿ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್, ಪಿಷ್ಟ, ಕೆಲವು ವೈರಸ್ ಅಲ್ಲಿ ಅಥವಾ ಕೆಲವು ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಇದ್ರೆ ಆ ಪಿಷ್ಟವನ್ನು ಮಾತ್ರ ಸಪರೇಟ್ ಮಾಡಿ ಅದಕ್ಕೆ ನಾವು ಪ್ರೋಟೀನ್ ಜೊತೆ ಅದನ್ನ ನಾವು ಲಿಂಕ್ ಮಾಡಿ ಅದನ್ನು ಕೂಡ ನಾವು ವ್ಯಾಕ್ಸಿನ್ ತರ ಎಲ್ಲ ಯೂಸ್ ಮಾಡಬಹುದು. ಅದ ನಂತರ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಈಗ DNA, ಕೇವಲ DNA ಕೂಡ ನಾವು ಪ್ಯೂರಿಫೈ ಮಾಡಿಕೊಂಡು ಕೂಡ ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. ಈಗ ನೀವು ಕೋವಿಡ್-19 ಇದರಲ್ಲಿ ಸಾರ್ಸ್ ಕೊರೋನಾ ವೈರಸ್ 2 ಈ ಒಂದು ವೈರಸ್‌ನಿಂದ ಅವರು ಕೂಡ ಏನ್ ಮಾಡಿದ್ದಾರೆ ಎಂಆರ್‌ಎನ್‌ಎ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಅಂತೆಲ್ಲ ಮಾಡಿದ್ದಾರೆ. ಅದು ನಮ್ಮ ಅದು ಫೈಜರು ಮತ್ತೆ ಒಂದು 2, 3 ಕಂಪನಿಯವರು ಕೂಡ ಎಂಆರ್‌ಎನ್‌ಎ ವ್ಯಾಕ್ಸಿನ್ ಅಗೇನ್ಸ್ಟ್ ಕೋವಿಡ್-19 ಅದನ್ನು ಕೂಡ ವೃದ್ಧಿಗೊಳಿಸಿದ್ದಾರೆ. ಅದಕ್ಕೆಲ್ಲ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ದೆನ್ ರಿಕಾಂಬಿನೆಂಟ್ ವ್ಯಾಕ್ಸಿನ್ ಇಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಯಾವ ಒಂದು ನಿಮ್ಮ ಆಂಟಿಜೆನ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರುವಂತಹ ಒಂದು ಸಣ್ಣ ಜೀನ್ ಅನ್ನ ಇರುತ್ತೆ, ಅದನ್ನ ಕ್ಲೋನಿಂಗ್ ಮಾಡ್ತಾರೆ. ಅದ ನಂತರ ಕಂಪ್ಲೀಟ್ ಕ್ಲೋನ್ ಅನ್ನೆಯ ಆ ದೇಹದಲ್ಲಿ ಚುಚ್ಚಿದ ನಂತರ ಆ ಡಿ-ಎನ್-ಎ ದಿಂದ ಎಂ-ಆರ್-ಎನ್-ಎ ಆಗುತ್ತೆ, ಎಂ-ಆರ್-ಎನ್ ಇಂದ ಪ್ರೋಟೀನ್ ಆಗುತ್ತೆ. ಆ ಪ್ರೋಟೀನ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಆಂಟಿಜೆನ್ ವಿಚ್ ಇನ್ ಟರ್ನ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಟ್ರಿಗರ್ ದ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್. ಅಂಡ್ ದೀಸ್ ಆರ್ ಆಲ್ ದ ಡಿಫರೆಂಟ್ ಮೇಜರ್ ಟೈಪ್ಸ್ ಆಫ್ ದ ವ್ಯಾಕ್ಸಿನ್ಸ್. ಈಗ ನಮ್ಮ ಲಸಿಕೆಗಳಲ್ಲಿ ಕೂಡ ವ್ಯಾಕ್ಸಿನ್ಸ್‌ಗಳು ಕೂಡ ಫಸ್ಟ್ ಜನರೇಷನ್, ಸೆಕೆಂಡ್ ಜನರೇಷನ್, ಥರ್ಡ್ ಜನರೇಷನ್ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಅಂತೆಲ್ಲ ವಿಂಗಡಣೆ ಎಲ್ಲ ಮಾಡಿದ್ದಾರೆ. ಈಗ ಫಸ್ಟ್ ಜನರೇಷನ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂದ್ರೆ ಅದನ್ನ ಪ್ರಾಥಮಿಕವಾಗಿ ಯಾವಾಗ ಒಂಥರ ನಮ್ಮ ವ್ಯಾಕ್ಸಿನ್ ಬಗ್ಗೆ ಎಲ್ಲ ನಮಗೆಲ್ಲ ಒಂದು ವಿಚಾರ ಎಲ್ಲ ತಿಳಿದು ಬಂತು, ಆ ಸಂದರ್ಭದಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡಿರುವಂತಹ ವ್ಯಾಕ್ಸಿನ್ಸ್‌ಗಳಿಗೆಲ್ಲ ನಾವು ಫಸ್ಟ್ ಜನರೇಷನ್ ವ್ಯಾಕ್ಸಿನ್ ಅಂತಾನೆ ಹೇಳ್ತೀವಿ. ದೇ ಆರ್ ಆಲ್ ದ ಕನ್ವೆನ್ಷನಲ್ ವ್ಯಾಕ್ಸಿನ್ಸ್. ಅಂಡ್ ಬ್ರಾಡ್ಲಿ ದೇರ್ ಆರ್ 2 ಟೈಪ್ಸ್. ಒನ್ ಈಸ್ ದ ಇನಾಕ್ಟಿವೇಟೆಡ್ ಆರ್ ದ ಕಿಲ್ಡ್ ಪೆಥೋಜನ್ ಅಂಡ್ ದೆನ್ ದ ಅದರ್ ಒನ್ ಈಸ್ ಲೈವ್ ಅಟೆನ್ಯೂಯೇಟೆಡ್ ಪೆಥೋಜನ್. ಇದ್ರ ಬಗ್ಗೆ ನಾನು ಆಲ್ರೆಡಿ ತಮಗೆ ವಿವರವನ್ನು ಕೊಟ್ಟಿದ್ದೆ. ಈಗ ಅಟೆನ್ಯೂಯೇಟೆಡ್ ಹೋಲ್ ಅಂತ ಹೇಳಿದೆ. ಇದ್ರ ಬಗ್ಗೆ ಕೂಡ ನಾನು ಆಲ್ರೆಡಿ ತಮಗೆ ಒಂದು ಸಂಕ್ಷಿಪ್ತವಾಗಿ ಈ ಹಿಂದೆನೆ ತಿಳಿಸಿದ್ದೆ. ಅದರಲ್ಲಿ ದೇ ಆರ್ ಆಲ್ ದ ವೀಕೆಂಡ್ ಮೈಕ್ರೋಬ್ಸ್. ದೇ ಕೆನಾಟ್ ಇಂಡ್ಯೂಸ್ ದ ಡಿಸೀಸ್, ಬಟ್ ಸ್ಟಿಲ್ ದೀಸ್ ಮೈಕ್ರೋಬ್ಸ್ ಹ್ಯಾವ್ ದ ಪೊಟೆನ್ಷಿಯಲ್ಸ್ ಟು ಅಂಡರ್ಗೋ ರಿಪ್ಲಿಕೇಶನ್ ಆರ್ ಪ್ರಾಲಿಫರೇಷನ್ ವಿದಿನ್ ದ ಬಾಡಿ ಆಫ್ ದ ವ್ಯಾಕ್ಸಿನೇಟೆಡ್ ಇಂಡಿವಿಜುವಲ್. They cannot cause the disease, but nevertheless, these attenuated whole agent vaccines can trigger the immune system. And these vaccines, they closely mimic the actual or the natural infection, but it doesn't induce the disease, unlike natural infection. What are the advantages of these attenuated vaccines? ಇದರ ಲಾಭಗಳಲ್ಲಿ ಏನೇನು? Attenuated vaccine ಅಂತ. Single dose is sufficient to induce the long lasting immunity. Single dose is sufficient. Then there is very, very strong immune response. Then local as well as systemic immunity is also produced. But what are the disadvantages of these attenuated vaccines? As I already told you, attenuated vaccines, they possess viruses or bacteria which still have capability to undergo replication or proliferation after they are injected into the host system. Maybe it is human beings or animals. But the disadvantages of these attenuated vaccines are there is a chance of reversion of these attenuated pathogens to their original virulence. This is very, very risky. Once ನಾವು ವ್ಯಾಕ್ಸಿನ್ ಅಂತ ಮಾಡುವಂತದ್ದು ನಮ್ಮ ಪರ್ಪಸ್ ಏನು? ಒಂದ್ಸರಿ ದೇಹದಲ್ಲಿ ಒಂದು ವ್ಯಾಕ್ಸಿನ್ ಆಂಟಿಜನ್ ಹೋದ ಕೂಡಲೇ ಅದು ನಮ್ಮ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಅನ್ನ, ನಮ್ಮ ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿಯನ್ನ ಜಾಸ್ತಿ ಮಾಡಬೇಕು ಅಂತ. ಅದ್ರ ಬದಲಿಗೆ ಅದು ವಿರುಲೆನ್ಸ್ಗೆ ವಾಪಸ್ ಹೋಗ್ಬಿಟ್ರೆ ದಟ್ ಮೀನ್ಸ್ ಇಟ್ ಕ್ಯಾನ್ ಕಾಸ್ ದ ಡಿಸೀಸ್. This is very risky. This is one of the disadvantages, and they are not that stable, and there is a possibility or potentials for contamination of these attenuated vaccines. What do you mean by inactivated whole agent vaccines? Here, as I've already told you in the previous slides, whether it is microbial, whether it is a bacterial or viral vaccine, they have been killed. That means they are treated with some chemicals or they are heat treated so that once these viruses or bacteria they are injected into the body, it cannot undergo multiplication. That is the meaning of these inactivated vaccines. But nevertheless, even then, these inactivated vaccines, these inactivated pathogens, they can still trigger the immune response, the immune system. What are the advantages of these inactivated vaccines? They are stable. There is absolutely no reversion to the virulence because they are killed. This is one advantage of killed vaccines over the attenuated vaccines. And they cannot cause the infection. That is the major advantage. But what are the limitations? What are the disadvantages? Unlike live attenuated vaccines, here the killed vaccines have to be repeatedly administered. ಇವತ್ತು ಕೊಟ್ಟ ನಂತರ ನೆಕ್ಸ್ಟ್ ಇಯರ್ ಕೊಡಬೇಕು, ಅದ್ರ ನೆಕ್ಸ್ಟ್ ಇಯರ್ ಕೊಡಬೇಕು, ಬೂಸ್ಟರ್ ಡೋಸ್ ತಗೊಳ್ತಾನೆ ಇರಬೇಕು. ಒಂದು ವೇಳೆ ನಮ್ಮ ಹತ್ರ ಲೈವ್ ಅಟೆನ್ಯೂಯೇಟೆಡ್ ವ್ಯಾಕ್ಸಿನ್ ಕೊಟ್ರೆ ಅದಕ್ಕೆ ಬೂಸ್ಟರ್ ಡೋಸ್ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಒಂದು ವ್ಯಾಕ್ಸಿನ್ ಕೊಟ್ಬಿಟ್ರೆ ಸಾಕು, ಜೀವಮಾನ ಪರ್ಯಂತ ಅವರಿಗೆ ಒಂಥರ ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿ ಕೂಡ ಇರುತ್ತೆ, ಅಥವಾ ಹಲವಾರು ವರ್ಷಗಳವರೆಗೆ ಆ ಒಂದು ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿ ಕೂಡ ಇರುತ್ತೆ. ಇದಕ್ಕೆ ಅಡ್ಜುವಂಟ್ ಅಂತ ಒಂದು ಬೇಕಾಗತ್ತೆ. ದಟ್ ಈಸ್ ಅನದರ್ ಅಡ್ಜುವಂಟ್, ಅನದರ್ ಡಿಸ್ಅಡ್ವಾಂಟೇಜ್ ಆಫ್ killed vaccines or inactivated vaccines. These adjuvant are something like some oil, it can be some oil or some other chemical which will effectively trigger the immune system in addition to the pathogen incorporated in the killed vaccine. Then another limitation of this killed vaccine is they induce the shorter immunity. They cannot induce the longer immunity, unlike these live attenuated vaccines. Shorter lasting immunity is induced by the killed vaccines. And the cell mediated immune response, CMI stands for cell mediated immune response, triggered by these killed vaccines is very, very poor. That is about the first generation vaccine. Now coming to the second generation vaccine, here we have what are called subunit or the peptide vaccines. I already told you that. See, some parts of these vaccines, this virus or the bacteria can be separated from this virus or the bacteria, and then it can be used as a subunit vaccine. Peptide means in case you take some protein out of the envelope or the capsid of this virus, and then you can use that independently as a peptide vaccine. What are the advantages? There is no risk and there is no DNA in this vaccines. But the disadvantage is that is protein the same as in situ? The proteins that you get out of such viruses or bacteria to be developed as a peptide vaccines, the protein or the peptide must be the same as that of the virulent bacteria or the virulent virus, which may subsequently infect the host system. Subunit or peptide vaccines are generated out of taking out a small part of the complete virus or bacteria, and it is not going to induce the disease because it is not the complete bacteria or the virus, and this is affiliated to second generation vaccine category. I already told you what is a toxide vaccine. Tetanus toxide anta heltivi. This is an inactivated toxin. As such, the toxin can induce the disease, whereas the toxoids cannot induce the disease. Toxides are nothing but the inactivated form of the toxins. What is a conjugate vaccine? What is a conjugate vaccine? As I already told you, there are some type of vaccines which use polysaccharides, the carbohydrates. And you know, some of the bacteria, they have these polysaccharide capsules. You have heard of what are called capsules. But unfortunately, these polysaccharide based antigens, the vaccines, they are very, very poorly immunogenic. And hence, to make them more immunogenic, these polysaccharides, they are combined with another protein to make the vaccine. And this is carbohydrate, and then it is linked with some other protein which is known as carrier protein, and both together can be triggering the immune system. And such vaccines are known as the conjugate vaccine, because here we have conjugated the carbohydrate with the protein, and this is the structure of the bacteria, which is actually covered by the polysaccharide coating. This is in a natural bacteria, and this is how we can take out these polysaccharides, link with the protein, and then this can be the conjugate vaccine. Yeah, this is about a second generation vaccine. So is it okay? Am I audible, and whether the slides are moving? One of you, please. Dr. Ashmi.
Sir, it is audible, sir.
ಸರ್ ಸ್ಲೈಡು ಕೂಡ ಕಾಣ್ತಾ ಇಲ್ಲ ಇದೆಯಲ್ವಾ, ಚೇಂಜ್ ಮಾಡ್ತಾ ಇರುವಂತದ್ದು?
ಕಾಣಿಸ್ತಾ ಇದೆ ಸರ್.
Okay, okay, okay.
Yes.
But
Yes.
but this is about second generation vaccine, the conjugate vaccines or the subunit vaccines or peptide vaccines. But still there is lot of need for further improvising the quality of the vaccines. That was actually accomplished through the third generation vaccines. And but what was the need for us to improve these vaccines? Because genetic variation is leading to evolution of different type of strains, and we have, all of us have experienced that in this COVID-19. We myself have actually seen several, at least 3 to 4 or more than 4 strains of SARS coronavirus 2. Vaccine against one strain may or may not confer effective protection against another strain. That is attributed to the genetic variations of these viruses. If there is a genetic variation or if there is emergence of different strains of viruses, then the vaccine developed to the earlier strain will not confer protection, effective protection against the subsequently responsible strain. And this is attributed to very high rate of spontaneous mutations in these viruses. And that was also occurring in the spike protein of the SARS coronavirus too. And vaccines are generally effective in controlling only clinical signs rather than infection. You know what I mean? So once we are vaccinated, it doesn't mean to say that we should be 100% protected against the infection. It will suppress the severity of the disease. That's also most of us have also seen, including myself. First, I had even before I took my first shot of a COVID-19 vaccine, I picked up a COVID-19 infection. After that, I have undergone vaccination. Second time, again, I picked up a COVID-19 infection. But second time, the disease was not that severe. Of course, during the first infection itself, it was not a severe disease. It was moderate. But in the second time, when I was infected with this COVID-19 virus, it was not at all. It was very, very mild. And that could be that mild nature of this COVID-19, which I myself have actually gone through, could be attributed to vaccination. Vaccination may not totally prevent from being infected, but it significantly it can reduce the severity of the disease. And then there is better price affordability, and we need to further make them more say cost effective, and it should be more efficacious so that several strains can be targeted by using a common vaccine. And then the durability of this vaccinal immune response, it should be prolonged. And for this purpose, we have what are called the viral vectored vaccines, and then adjuvants are being used along with the inactivated vaccines. And one of the limitations of these inactivated vaccines, because it is administered along with some chemicals to effectively trigger the immune system, those chemicals are known as the adjuvants. And then the adjuvants, deposition of adjuvant at the site of injection, it has resulted in development of small nodular lesions, so small sarcomas, and which most of the vaccinated individuals, they find it uncomfortable. That's the reason. That's one of the reasons why the adjuvanted vaccines may not be preferred by some of the individuals. And all these limitations of the first generation vaccines as well as the second generation vaccines have actually made the scientific fraternity across the globe to seriously think of developing the third generation vaccines, and hence at present it is known as an era of novel vaccines. What are these third generation vaccines? And this comprises of the DNA vaccine, and these DNA vaccines are nothing but the genetically engineered vaccines which can still trigger the immunological response in the host. And we have the DNA vaccines against bacteria, parasites, viruses, as well as there are DNA vaccines against some of the potential cancer conditions. How these DNA vaccines are made? You know, first we have the gene. I'm sure all the participants are aware of the DNA, which when undergoes transcription makes the mRNA. Then mRNA will undergo translation to make protein, and that protein is nothing but the antigen, nothing but the source of the vaccine. Yeah, with that in the background, let us see how this DNA vaccines actually work. You see, this is a double-stranded viral gene, and this is cloned into what is called a plasmid. Then the ends are tried. Now this viral gene, which is going to express the protein or the antigen of your interest, is cloned. Cloned means it is inserted into the plasmid. Plasmid is again nothing but the double-stranded DNA. Then the ends are tied, and then it is introduced into the bacterial cell. Once it is introduced into the bacterial cell, then the bacteria undergoes multiplication. One bacteria may lead to 4 bacteria, but each of the 4 daughter bacteria, they will carry this clone containing the gene of your interest, which when undergoes transcription translation leads to expression of a protein. In turn, that is nothing but the antigen that in turn will trigger the immune system. Yeah, this is the plasmid, and then straight away the plasmid is actually injected into the body of the. Then it will enter into the skin. Instead of a bacteria, it can just enter into the skin, and then it is taken up by macrophages, and then it is processed, presented to the T cells, B cells. B cells will produce the antibodies, then it will neutralize that, whereas the T cells will transform into CD8 killer T cells. These killer T cells will actually go and eliminate the cells which have been infected by the virus in the subsequent episodes. This is how basically the DNA vaccine actually function. But how these DNA vaccines are delivered? There are different jet injectors, there are gene guns, and there are injections, and then even by electroporation also, these DNA vaccines can be delivered set at different layers. You can just surf through these pictures. Even microneedle technology is also becoming very popular. Just like a patch, you will have microneedles which are actually studded with these DNA vaccines, and then you have that patch on the hand, and then gradually it will this which will have microneedles. It's not that painful because it is just a microneedle, unlike the needle which is used for injection purposes. And the microneedle at this portion, it is actually uh, containing the DNA. Once it is actually introduced into this dermis layer, it will deliver the DNA vaccine into the tissues. Then the DNA will undergo transcription to make messenger RNA. Messenger RNA will undergo translation to make against this particular bacteria. You can take this as the genome of the bacteria, clone it into the plasmid DNA, and then you inject this plasmid DNA. Then blue color is the gene collected from the genome of the bacteria against which you would like to develop the vaccine. Then the whole thing is injected into the human body, and then it will undergo transcription within the human body, undergo translation. Then these proteins are expressed on the surface vaccine. Because of the time constraint, I may not be able to throw all that, but these DNA vaccines are reported to be relatively inexpensive, and the structure and post-translational modification of antigen is like in the natural say infection. Whereas as far as the disadvantages are concerned, there is no mass application methods for animals. The efficiency of these DNA vaccines is relatively less than compared to the already established vaccines. Then I'm going to share this PPT with the KSTA authorities, and then all the participants can have access to this. Then coming to what is RNA vaccine. Just like DNA vaccine, we have RNA vaccines. These RNA vaccines are nothing but the mRNA vaccines. And this is actually constituting a major chunk in this new era in vaccinology. I'm sure all of you have heard of these mRNA vaccines in the pandemics of COVID-19. You know, the Pfizer as well as Moderna COVID-19 vaccines, they have made this mRNA vaccine. So what they do is, here instead of a DNA, it's only the mRNA from the organism. So which when introduced into the host cell, you see this is SARS-CoV-2, and then the mRNA from this SARS-CoV-2 is injected into the body. Then this mRNA will undergo translation to make spike proteins, because this mRNA was isolated from SARS-CoV-2, which in turn is going to express the spike protein. These spike proteins are taken up by these antigen presenting cells. It is processed, and then it is presented to the T cells, and I had already described about these major steps. These T cells will undergo transformation to make T cells, which CD8 cells, which will destroy the cells. The B cells are generated. B cells will lead to generation of plasma cells. Plasma cells in turn will make the antibodies. These antibodies will neutralize the virus. This is how the mRNA vaccine actually functions. There are certain challenges with these mRNA vaccines. You see, the mRNA, once when injected into the body, can be neutralized, or it is not that stable. It undergoes degradation very fast. Then storing of mRNA for a long time is a major challenge. This is one of the reasons why the Pfizer vaccines and then Moderna vaccines against the COVID-19, they did not become popular in most of the tropical countries, because to preserve mRNA vaccines and then ship it to villages and different places, in India or different most of the tropical countries, they needed to have the deep freezers. We are not having so much of infrastructure as far as preserving this mRNA in the deep freezing conditions are concerned. That is the reason why these mRNA based vaccines, because of their stringency in preserving them at very low temperature, so they did not become successful. Now, what are these vectored vaccines? Viral vectors, viruses, the genome of the virus itself is actually used as a vector. Instead of a plasmid, you can use the genome of a virus itself as a vector. Then you can clone the gene of your interest, which when undergoes transcription translation will make the antigen or the protein. So likewise, you have heard of this Covishield vaccine, isn't it? COVID-19, Covishield, which is again prepared by this Serum Institute of India in Pune. They have used adenoviruses as the vectors. What they have done, they have cloned the spike protein expressing gene into these adenoviruses, and then they have used that as a source of vaccine. And this is where the vector vaccines are actually playing a major role. This is just the illustration how exactly the vector vaccines actually function. You see, this is again yet another plasmid, and you can clone the DNA coding for antigen from the pathogen. Okay, then in this picture you can see here it is cut in the plasmid. This DNA encoding antigen from the pathogen is introduced. Then you take the tissue culture monolayer cells. You super infect with both recombinant plasmid, which is containing the gene from the pathogen, which is going to code the protein to confer protection against this pathogen, along with the vaccinia virus. The cells are infected with both recombinant plasmid containing the gene of your interest as well as the vaccinia virus. Now there is homologous recombination. Now what happens is this vaccinia virus, which is again a pox virus, this is not harmful, this is not pathogenic, or this is not virulent. Once the tissue culture cells are infected by this vaccinia virus, at same time this is infected, this is transfected by recombinant plasmids. Then what happens? Here you see this is the genome of vaccinia virus. Here this is the recombinant vaccinia vectored vaccine. You see the difference between this and this. This is actually having the gene of your interest, the red colored, which is actually picked up from this recombinant plasmid. So once both these recombinant plasmid and vaccinia virus, they infect the same cell culture system, then because of homologous recombination between these two, this red gene from pathogen will jump into the genome of the vaccinia virus, and then the vaccinia virus can be used as a source of vaccine. It can be used to inject. This is what precisely was done as far as the development of Covishield vaccine against COVID-19 is concerned. Yeah, this is about the RNA viruses. The same strategy is applicable. RNA virus, then you can purify the RNA. Using the RNA, you can make the complementary DNA and then infect the cell with this complementary DNA as well as the pox virus. Pox virus undergoes genetic homologous recombination, and then the pox viral genome will pick this cDNA of this particular virus, maybe a rabies virus. Then the viral antigens are expressed. Because this vaccinia virus, it is used as a source of vaccine. Then it will undergo the recombinant DNA present in the vaccinia virus will undergo transcription translation, leading to expression of the protein of interest on the surface of the target cell. Then in turn this is going to trigger the immune response. This is how the RNA viral nucleic acid can be used to develop these viral vector vaccines. Now, what are the marker vaccines? This is very, very important. See, supposing some animal, it gets infected with foot and mouth disease, FMDV, then it will definitely lead to development of antibodies to FMDV virus. But here it is, the natural infection means the animal is going to be infected with the whole virus. You see, this is green colored is the virus. This is called the structural protein, green colored, hexagonal shaped. Whereas you can see some spiral red colored structures. These red colored structures are nothing but the enzymes. You know, enzymes are necessary for the replication of the virus in the natural infection. So structural protein which makes the virus is also protein, whereas enzymes are also proteins. So when the immune system is actually exposed to this complete infectious virus, then the antibodies are developed against the structural proteins as well as antibodies are developed against the enzymes. Against the enzymes. So here you can see antibodies against the structural proteins, that is green colored protein. Then antibodies are developed against non-structural proteins. These NSPs are nothing but the enzymes. Whereas if you develop a marker vaccine is one which does not have the NSP component. Here you see this is only the structural protein, only the green protein. Only this is the virus without the non-structural proteins, without the enzymes. Whereas here this is naturally existing FMD virus which contains the structural proteins as well as the non-structural proteins, the red colored ones. So in turn it leads to generation of antibodies to structural protein as well as NSPs or non-structural proteins, enzymes. Whereas in case of these marker vaccines, it is only the structural proteins because they are generated in the lab. Then once it is injected as a source of vaccine against FMDV, then antibodies are generated only against the structural proteins, but there are no antibodies against the enzymes or the NSPs because this vaccine does not have the NSPs or the enzymes. And that is one major difference as far as the triggering of immune system by FMDV virus from the field as well as those vaccinated using the marker vaccine against FMDV. So if you take these say serum samples from such animals and then test them, you will have only structural antibodies to only structural proteins. Whereas if you collect the serum sample from these animals which are naturally infected by this FMDV virus, FMDV, then you will have antibodies to both enzymes as well as the structural proteins. So the presence of antibodies to enzymes in infected animals helps us to differentiate the immune response, whether it is because of vaccination or because of natural infection. Because in both the systems you will have the antibodies. But how will you differentiate whether the antibodies are because of vaccination or because of natural infection? By employing this marker technology, it is possible to differentiate because you do not have antibodies to NSPs in the animals which are vaccinated using the marker vaccines. I am pretty sure this concept is very clear. Now, coming to the oral rabies vaccines, these oral rabies vaccines are becoming very, very popular. You see how important rabies in case of dogs is concerned. We have also discussed that during the visit of all the participants to our rabies lab is concerned. Dogs are the major vectors as far as transmission of rabies to other animals or human beings is concerned. How do we need to control rabies in dogs? You need to catch the dogs and then catch them and then you inject these vaccines. But catching the dogs, especially the stray dogs, is a Herculean task. If somebody can catch the dogs, then injecting is not at all an issue. Then another issue is how are we going to control rabies in jackal? How are we going to control rabies in wolf, in mongoose, in the wild animals? You cannot simply go and enter into the forest and then catch these jackals and wolf. They are wild animals. And recently, just last week itself, we have reported one more case of jackal rabies near Harihar Taluk in Davangere district. Just we reported it just last week, Srinivasji. So, but how is that we are going to tackle these this deadly disease rabies in case of wild animals as well as street dogs? Here we have the concept of using oral rabies vaccines. What we do is the vaccine is incorporated into the baits. Some meat or some egg yolk is used as a bait, BAIT. You know what is a bait. Within that you introduce the vaccine containing sachet and then throw it onto the street. And once these dogs, the street dogs or the wild animals in the forest, if it is thrown, if it is distributed through helicopters, then they can consume it. They will be immunized against rabies. And because this is a live attenuated vaccine, oral rabies vaccines, it will trigger the immune system of these uh, animals which consume oral rabies vaccines. They are protected for life against rabies. So here you can see some pictures. You see they're all oral rabies vaccine containing sachets, and through helicopter they can distribute it, they can throw it into the forest area where we have large number of wild carnivores. They consume this and then they develop immunity against this. And this is what we would like to introduce to control rabies in the street dogs even in India. In fact, I have been very closely monitoring this particular situation, wherein we have actually convinced the government of India to permit us to import this oral rabies vaccine from different countries, because as of now we are not using oral rabies vaccine in India, and soon we are going to have this oral rabies vaccine introduced in India, so also in Bangalore And then we are going to use this oral rabies vaccine for control of rabies in street dogs. You can see the size of this oral bait rabies vaccine. This is one wild animal, skunk, and the coyotes. They play a major role in transmission of rabies in the United States. You see, it is a throne, and it is holding, and then it is consuming the vaccine. These are all different types of vaccines which are available for animals against different viruses. I can say some more vaccines which are used in animals. We have pox viral vaccines, sheep pox, goat pox, and then Marax disease in case of a poultry, Newcastle disease in case of a poultry. Then we have swine fever in case of pigs. Then avian infectious bronchitis, again caused by coronavirus. We have in fact coronavirus infections lurking in poultry population. That is called infectious bronchitis. And regularly we have been vaccinating the poultry population against this coronavirus infection. Thank God this coronavirus infection infecting dogs or cattle, bovine coronavirus or porcine coronavirus or the poultry coronavirus, they are not infecting the human beings. They are entirely different from the SARS coronavirus 2. You see here the different brands of COVID-19 vaccines have been used. This is mRNA vaccine, Pfizer BioNTech, and this is Covaxin, which is inactivated vaccine. And then we have a couple of other vaccines from different sources. So to conclude, most of the infectious diseases, they can be prevented by employing timely vaccination. You know, pulse polio vaccination programs have become very, very successful, very, very popular. The only thing is that the general public must cooperate with the government schemes to control these diseases and then come forward to take the benefits of this vaccination. And let us always go ahead with the slogan that prevention is always better than cure. So let us promote the effective vaccination strategy so that people need not have to really suffer from some of these deadly disease and then think of going for antibiotics or the antivirals, which at times may not work. So keeping this in view, we go with the strategy of prevention is always better than cure. So with that, I just close this talk. I will be happy to invite some questions. Thank you, KSTA. Thank you all the participants for being there with me. And thanks to Professor Ayyappan sir, Dr. Ramesh sir, Dr. Mr. Srinivas sir, and then Mr. Anthony, and then all the members of the KSTA family. Thank you. Elrigu namaskara.
Thank you so much, sir, for your very much interesting lecture. Are there any questions from the participants? So at present, no questions, sir. So if there are any questions, we will write back to you, sir.
Oh, sure, sir. Definitely.
Yes, sir. Thank you so much, sir, for your
Okay, sir.
excellent lecture, sir. Thank you, sir.
Oh, ugly, sir. Thank you so
Yes,
much.
sir. Yes, sir. Now we have Professor Acharya sir talks, sir. Namaskara, sir.
Good afternoon, everyone. Namaskaram.
Namaskara, sir. So good evening to one and all again. Today we have the last session of from Professor Kritish K. Acharya, sir, presently Professor Transcriptomics and Reproductive Biology at Institute of Bioinformatics and Applied Biology, IBAB, Bangalore. So Kshitish Acharya sir received his PhD from Banaras Hindu University, Varanasi, and completed postdoctoral research at IISc Bangalore, and then moved to University of Virginia, America, head of research and production units, Is Rna Biotechnology Limited during 2002, and founder of Biological Data Analyzers Association in 2015. At IBAB, Since early 2003, Servas has been trying to get new insights into the mechanisms of regulation of gene expressions in different systems under disease and normal conditions. Professor Acharya sir started his research career with wildlife studies in Western Ghat forest, then took the behavioral, histological, and psychological, physiological, and in vitro culture studies. During his postdoctoral work at the University of Virginia, America, he mainly applied recombinant DNA technology, cell culture, and other molecular techniques to characterize a promoter and cognate regulatory proteins. Sir research mainly used mouse as a model. Through he worked on other animals such as bats, hamsters, and bonnet monkeys. Sir worked on protein purification methods at Is Rna Biotechnology Limited in Mumbai. Main research areas: Kriti's group has been making combinatorial use of modern approaches in molecular and computational biology to study transcription control mechanisms, identify novel markers for male infertility and cancer of specific tissues such as prostate, cervix, liver, breast, and also lung. Addressing reproductive biology at the molecular level, Sir Group first painstakingly compiled gene expression data over multiple years, developed tissue-specific gene expression databases, testes, prostate, and uterus, and found ways to make a better use of existing data. They also developed some of the naval software and computational methods for the analysis of the resulting data to understand the regulatory mechanisms or experimental validations. So using RNA sequencing, Sir Group recently shortlisted markers for a type of male infertility and established a novel association of several genes with human spermatogenesis. Currently, they are trying to explore the role of some of the novel genes. Part of his team is also contributing to a nationwide project that explores the Indian human genome in the context of hereditary deafness. Apart from teaching, Sir is also guided and supervised a few PhD students and about 100 interns at IBAB. While he has been enjoying a multidisciplinary research apart from teaching in regular program, Sir has also been a visiting fellow and professor at University of Sydney, Australia, MCO PS Manipal, and also CHG, JNCSR, and IIT Bangalore, Triple ITB Bangalore. Sir is frequently volunteers to contribute to scientific seminars and educational programs, sometimes in Kannada too, particularly for school children across Karnataka. He also conceptualized, designed, and successfully convened a unique postgraduate diploma program in biotechnics and organized more than 25 specialized short courses for various types of at IBAB. Sir supervised and guided multiple projects related to metagenomics, genomics, transcriptomatics of plants and humans, as well as microbiomes from various sources at Shodhaka, Bangalore, a company he founded and also funded. At Shodhaka, Sir team also developed a multiple bioinformatics resources over 10 years and a bioinformatics global portal, that is www.startbioinfo.com, in association with Biological Data Analyzers Association. With this brief introduction about her, on behalf of all of you, Karnataka Innovation Technology Society, Department of IT, BT and SNT, Karnataka Science and Technology Academy, DBT, Government of India. So we a warm welcome to you, sir, giving the talk on bioinformatics. Sir. Welcome you, sir.
Thank you, sir. Thank you for a very kind introduction, and even more thanks to all the participants who kindly listened to all the details. It was not necessary. I felt a small introduction would have been enough. You have taken a lot of trouble, sir, all of you. So, but anyway, thank you. Let me get started.
Yes,
I have put the title as Bioinformatics, a Discussion. The reason is I would like to talk to you all, okay? And I just want to discuss, so feel free to ask any questions at any point during the presentation, and I'll be happy to discuss, because there is no point in me trying to tell whatever I know. Rather, because you are a variety of people from variety of background, so I'd like to keep it more in the discussion mode. So that's my first request to you. Rather than waiting for the end to ask questions, you can just pitch in anytime, comment or question, please. Okay. I hope I'm audible, by the way.
Yes, sir.
Yes, sir.
Okay, thank you.
Full screen will be much better, sir.
No, I just need to type things on the go, so I have kept it little.
Sir, you can use the whiteboard, sir. That option is available.
No, whiteboard would not have so much flexibilities, but
Yes,
let's see,
sir.
I will use big fonts, so please
Fine,
don't
sir.
worry.
Fine. Thank you, sir.
Yeah.
Please
So I would like to start by reminding all of you about what is research all about. So as biologists, we do, so I think the font sizes are big enough.
Yes, sir.
But if there is a problem, let me know, I can make them even bigger. So research leads to data and sometimes data and information, or only information. We all know about that. So I began my research career as a wildlife biologist. So we used to simply note what type of animals are there in the Western Ghat forests. And in those days, it was just like spotting an animal. Okay, where did I spot what animal, what type of animal, and what basis I am saying that this is the animal, etc. We used to write down. That itself is the data. So in the end, when we compile over a month's period, we noted, so we will have a sort of a map of the forest area we visited and where did we spot what type of animal. So this we used to record, and that is our data. So if you look at any disease also, so people, for example, suppose there is a strange disease that as people started noticing, and these people are having breathing difficulty, etc. So you do research on that. When you do research, okay, what type of food you took, what was the genetic background, did your parents have this disease, etc. You ask questions and you get the data, you get the information. And of course, days changed, and then people started asking more serious questions. They can actually take the blood under microscope and study as times changed. So different, more type of information started coming in. So we are all familiar as researchers, the type of data and information we need to accumulate and analyze them. So when you accumulate the data and analyze them, we get information most of the times. But what has changed over a few centuries, particularly in few last about 70, 80 decades, is the type of data we have changed. That is because of the type of technology. And this is, in other words, directly related to the type of technology and previous knowledge that was available. Okay. So these things made a difference. They are sort of reciprocally, influencing each other. So we know that. That has led to this field of bioinformatics. That's the nutshell of what I wanted to tell today. Okay. And what are some of the activities people do in bioinformatics, and how can it be useful to you or your students is what I would like to discuss today. Now, may I know what is the background of the participants? You're all teachers, I'm presuming, and teachers and researchers perhaps from different field.
Yes, sir. All are sir UG, PG faculty, and some of them are research scholars, sir. All are life sciences background, sir.
Life sciences, and so I'm presuming zoology, botany, microbiology,
Yes, sir.
biochemistry.
Yeah, yes, sir. Exactly, sir.
So, so I'm going to make a jump now. I will not get into too much details what type of technology change the type of data we are getting, etc. But we all know when we look at any type of research, for example, there is a disorder, let's say we start with the way we used to earlier address the issues, what could be the reason for this and what is the mechanism for it. The things changed because of the technology. First thing came out is the microscopy, and then there are a lot of other techniques that came. So today we know everything that happens inside our body is basically coming from the activities inside the cells. So that's why there is a reason why we define cell as the fundamental unit of life. because there is nothing living without a cell. And there is no, even if you look at multicellular organisms, all the cells that we have started from a single cell, but eventually every tissue has so many cells, they do different functions, etc. We know that. But inside a cell, it is the molecules that actually do the functions. We know that, right? So molecules make the cells. We sort of have a very clear idea of this. And you also know what is the relationship between different type of molecules and their responsibilities. Okay, so with this awareness, we also know most of the research questions that we asked today about diseases in human beings or animals. Just now we were talking about rabies, for example. So rabies is a virus. So what is the nature of the virus? How does it, where does it bind, to what type of cells and what type of proteins it binds, etc. How do we develop vaccine? All this is molecular biology. So that's the same thing with the plants also. When you look at agriculturally important plants and how do we modify, how do we get drought resistant plants, for example, people are now trying to deal with molecules and try to understand more about them. Either it's simply distinguishing the different varieties of the plants, or finding genetically engineered plant, which may or may not be allowed in different countries, but that seems to be the major hope for the world and for future. So addressing the molecules, studying the molecules has become one of the key areas of research. And this has started long back. So, you know, in 1950s, the DNA structure was found, and people knew by that time that genetic information is carried by the DNA, which means the nucleic nitrogenous bases. So the sequence of, so the sequence of nucleotides, if I may just write randomly some sequence here, this is how the DNA, if I write one single strand of the nucleotide, so maybe it will go like this. Right? It could be anything like this. So the sequence is what is most useful for the cell, because that's the genetic hereditary information, and that is converted to the sequence in the RNA molecule, and that in turn is used to produce the sequence of amino acids in the proteins, and proteins automatically take a function and they perform the particular, they take a particular shape and they perform a specific function automatically. Obviously, the sequence became a new type of data. Sequence, what was the new type of data that led to the need for computational biology I'm trying to mention now? Sequence, then structure, then interactions. Okay, interactions among the molecules. This type of data was never existing before. And to handle this type of data, I mean, you know, the proteins are the molecules which perform the functions in the cells. If the cells are the tiny factories that make the life possible, if cells are the tiny factories that make the life possible, it is the proteins which are actually the nano machines that perform most of the functions, whether simple thing like oxygen being carried out by hemoglobin, or actin binding to myosin, and then hence muscle action is possible, or a protein on the muscle is actually receiving a signal and then contracting something else. Like all these functions are carried out by proteins. So proteins are the laborers or the nano machines which perform functions automatically. But the protein production information lies in the DNA somewhere. So we have an idea of this now, but if you look back in the time when sequences started generating, so again in 1950s this happened. People, there were Frederick Sanger got the Nobel Prize for Sanger sequencing method, and that's when we started slowly generating sequences of genes, etc. And that type of data was too difficult to handle. Now, if I just take this sequence itself, let me duplicate this slide, and let me ask you a question. A small test, let's say. I will say, find a sequence such as GT in the sequence. Will you be able to find it? It will take some time, but you will be able to find, I guess, right? Find GCAAGT in the entire sequence. So you will start looking for the sequence somewhere here. Is it there, etc., etc., and then you will find out, okay, it's here. You need to find sequences because sequences are the ones which carry specific information. So it was easy for us to do this small exercise. But how about finding about 1,346 base or nucleotides? So this is how we abbreviate it. Region, I have some new sequence now. I have a question. Is it? We try to find it in the database because I want to find out, is my sequence new or already known? If this is a question you have got by whatever experiment you have done, you have cloned something and you think this is important, 1,346 long nucleotide. Is it there in the database of nucleotides? So when I say database of nucleotides, how big is it? You find out any sequence, you deposit in databases these days. This has started long back. You can actually, finding this one in this sequence is one thing, but finding 1,396 nucleotides in a database of nucleotides which has, let's say, about million sequences, each of, let's say, 10,000 nucleotide long region. So 1 million into 10,000. Now, if I have to find out if this is already known or not, how much time it will take. So this is the type of challenge people faced. Obviously, you cannot do with this type of analysis and detection of sequences, etc., without computational help. That's where computational biology crept in, and that's what led to bioinformatics as a field to emerge. Okay, so if I can just shift gears, I hope you will be able to see my other slides also. So do you see my other slide now, or you're seeing the old one only? I shifted my slides.
Yes, sir. We are new slide, sir.
Thank you very much. So this is an actual sequence from a database. There is a particular structure to it. Okay, so this is the protein sequence I'm showing you. The amount of sequences started increasing because of the technology. I hinted in the beginning that there is a relationship between the technology and the amount of information and type of information. By 1982, how many millions of base pairs? So what you see here on the red is millions of base pairs. So you had less than 1 million base pairs deposited into the database in 1982. Okay. And how many sequences were deposited is also shown there in the axis here. You will see that around 1998, you started seeing a big jump. And by 2005 or so, you have 40 million sequences, each one having different length. Okay. So you have about 40 million sequences deposited. Yes, 40 million sequences deposited by 2005. And you can see the rate of increase. And how many billions of base pairs, how many billions of nucleotides were added, that is on this scale. So it's almost like 1,000-fold more than this. Each one is around 1,000 nucleotide. But of course, around by 2010, this scale itself was useless because it was always straight. It kept going up like this. So you cannot use this scale. Okay. So things changed drastically because the technology changed. The pace with which you can sequence, the amount of money required to sequence certain length, all that became more efficient. So cheaper and faster. So things became, now people started measuring in the petabytes of storage data required. This is around 2009, you can see. So around 20 petabytes was used, and people already calculated 70 petabytes of required space total. So the scale has completely changed. And now the challenges that I was mentioning, there are more challenges than the simple thing. So whatever I told here, this itself becomes pretty simple task now. But by the way, people have written software which can do this type of job in about, if you want to find out whether it is known or already known, you perhaps need less than 1 second time for the computer to find out. That's the speed with which the computers are operating with using the tool called BLAST. Many of you are familiar with it. But the challenges are increasing because in terms of what is the speed with which you should analyze the data, because there is so much more data on one hand. On the other hand, new types of questions are being asked. Okay, so this is what computational biology is about. So to store the data of huge quantity is one thing, like I was mentioning here. So they started working around this area, this time only. They started developing databases to store the data very carefully, systematically. On the other hand, they developed software to analyze this type of data on large scale. So this is one type of information and data management people in bioinformatics do. So it became a specialized field as bioinformatics around 2000. It actually started as a big field as bioinformatics as a major domain. So there were courses starting on it. There were PhD programs starting on it. There were departments in universities starting with bioinformatics, etc. All that started. More importantly, today we are in a time zone where no biology seems to be effective without bioinformatics. You don't have to do bioinformatics yourself, but you may have to collaborate with somebody to deal with biological information. That's the stage we are in. So I was telling what other types of information we deal with. I mentioned that the structure of molecules is also critical. So there are a lot of different type of structures we deal with. Don't know if I have any snapshots of the structures of molecules. I did not keep. I'm sorry, but yeah, you are, I'm sure you're familiar with protein structures of various types. And those structures are critical in analysis of the functions, because every protein, finally does its own job because of its function, and people are interested in structural studies. This is a small schematic representation of the growth of structures over time. The number of X-ray based structural studies, NMR experiment-based structural studies, electron microscopy-based structural studies, which is coming recently. This is getting more popular nowadays. So people have been, this is all still old data, 2016, 17 I'm talking about. The scale is still increasing. Particularly the electron microscopy-based structural studies are increasing nowadays much more. So because structure is so important for the protein function analysis, people have been studying structure also, and analyzing that information is even more peculiar. So when I look at structure of protein, what is the bioinformatics there? Let's take a peek into it. I try to give you an example of what we do in sequence analysis, right? So I gave you an example, like I have a sequence like this, you find the sequence. This is a very simple example I'm giving. There can be a lot more. For example, take the lot of entire chromosomal sequence and find out where are genes, where are regulatory regions, etc., etc. So there are a lot of questions people ask in bioinformatics. I am just giving one small example of sequence analysis. So sequence analysis itself is a huge area. So I'm trying to give you examples of the types of activities already. I hope you get that point. Okay, so types of activities in bioinformatics. So I was telling you what types of data that led to in generation of this field bioinformatics. I'm already telling you corresponding areas of activities. Sequence analysis is one thing. I told you a glimpse of that, to how people do it. Structural analysis, okay, computational structural biology you call. So this sequence is very little bit straightforward, whereas here what people do is they look at 3D location of each atom in each amino acid. So in the three-dimensional space, where are the atoms located? So which basically leads to the relative distance and influence across amino acids, where there is more space, etc., etc. Obviously, this has a lot of implication in identifying the function,